Hello everyone. So today we will learn about amino acid classification. The short trick I am providing you today will be beneficial for you if you are a student of class 12th, because I think the chapter of biomolecules contain this part of amino acid classification. If you are doing BSc, if you are a master student, it will be beneficial for you, because amino acid is a very basic topic which will be very beneficial for you if you are preparing for any other competition exam also. So there are some amino acids which are sulfur containing, and the example of the sulfur containing amino acids are cysteine and methionine. The hydroxyl containing amino acids are serine, threonine, tyrosine. So you have to learn these names because they will become in like every competition exam. I think the hydroxyl group containing amino acid is like very important. So the hydroxyl containing, which means I think OH containing amino acids are serine, threonine, tyrosine. Okay, then you have to know that the hydroxyl containing amino acids are very susceptible for phosphorylation. Okay, then we have amide containing amino acids, and the example of amide containing amino acids are asparagine and glutamate. They are asparagine and glutamate. Then we have indole containing amino acids, and the example of indole containing is tryptophan. It is tryptophan. Then we have carboxyl group containing amino acid. The amino acids which have carboxyl group are basically acidic amino acids. So they are aspartic acid and glutamic acid. Okay. Then we have aromatic amino acids. The example of aromatic amino acids are tryptophan, tyrosine, and phenylalanine. Then we have acidic amino acids. Acidic amino acids example is aspartic acid and glutamic acid. Basic amino acids, the example of basic amino acids is lysine, arginine, and histidine. That is KRH, lysine, arginine, and histidine. Then we have hydrophobic amino acids, and the example of hydrophobic amino acids are GAVLI, PMWF, glycine, alanine, valine, leucine, isoleucine, proline, methionine. Methionine is example of cysteine containing amino acid also. Okay, sorry, sulfur containing amino acids also. Okay, tryptophan, tyrosine. Then we have polar uncharged amino acid. The example of polar uncharged is serine, threonine, tyrosine, cysteine, and glutamate. Now I will tell you some basic, basic tricks for learning some amino acid, like some important points that I think it will be beneficial. So proline is an amino acid which is basically a helix breaker. So proline is also known as helix breaker. In proline, instead of an amino group, an amino group is present, which means in proline, amino group is not present. In them, amino group is present. Okay. In tryptophan we have indole ring. And the indole ring absorbs ultraviolet radiations at 280 nanometer. So it is very important that in which amino acid indole ring is present. So indole ring is present in tryptophan amino acid, and this indole ring absorbs ultraviolet radiation at 280 nanometer. Okay. Then the other trick I think which will be important, in methionine, the sulfur is present, but still the sulfur is present, it is not able to make disulfide bond. Cysteine have sulfur also, and it is able to make the disulfide bond, which means methionine and cysteine both have sulfur, but methionine is not able to make disulfide bond, but the cysteine is able to make the disulfide bond. Okay. Other than that, we have serinocysteine. Serinocysteine is not a derivative of serine, it is a derivative of cysteine. You have to learn this line, that serinocysteine is not derived from serine, it is derived from cysteine. Okay. Okay. Then we have some standard amino acids example, which are basically 20 plus 2. The 20 are the standard amino acids. other than 20, the two are also standard amino acid, and the example of two is selenocysteine and pyrrolysine. Selenocysteine and pyrrolysine, these two are the example of standard amino acids. Okay, like other than the 20, because 20 are basically the magic na, so we all know about 20, but the thing is ki we don't know about the other two, which is selenocysteine and pyrrolysine. Then you also have to know that in arginine, guanidinium complex is present. In arginine, guanidinium complex is present. You have to learn that. This is very important and it will be beneficial for all the BSc, MSc and the students which are preparing for competition exam. So guanidinium ring complex is present in arginine. Okay. And then in histidine, imidazole ring is present. Histidine, HI, I say you have to learn that in histidine, imidazole ring is present. Histidine act as a buffer solution at physiological pH. Histidine act as buffer solution. So if the question comes like which amino acid act as a buffer solution, so you have to mention that histidine act as buffer solution at physiological pH, and histidine has